this tissue, this is the newly lactating breast. It makes proteins that this body has never made before. And even though most of them go out into the outside world, some, like alpha-lactalbumin and beta-lactoglobulin, actually end up in the bloodstream. Right? And the cells making those proteins have MHC schleppers on the surface, which display peptides that are being made inside. So those cells are now looking foreign, and yet the new lactating breast is not rejected. And so my professors said, oh, that's because when this individual was learning self, she was drinking mother's milk and becoming orally tolerant, which we may have time to get back to. So I pointed out to them that my generation's actually done an experiment. Millions of women around the world after World War II didn't breastfeed. They fed Nestle's and pet milk and carnation milk. And yet, when we grew up and started lactating, our breasts didn't fall off. Somehow, the immune system knows how to deal with a changing self. That was one of the problems I had as a student. And since you guys, many of you are students, I want to tell you something that's really important. Right? If something confuses you, if something doesn't make sense, even if it's been in the textbooks for 50 years or 100 years, question it. If it doesn't make sense to you, dig your professors in the side and say, this doesn't make sense. Because you'll be surprised at how much absolute bullshit there is written in textbooks. Okay, so that was one of the problems I had. Here's another one. This was also a problem for a guy named Charlie Janeway, who also questioned that model. And that is when you're trying to make a vaccine, if the vaccine is an attenuated virus or a dead bacterium, usually just injecting that vaccine is enough to get an immune response. But if you're trying to make a modern vaccine, a peptide vaccine or a synthetic protein vaccine, people have found that they inject them, even in large quantities, and nothing happens unless you add something called adjuvant. Right? So for example, if you inject egg white protein, ovalbumin, into a mouse, which is definitely foreign to a mouse. Right? It doesn't matter if you inject it intravenously, intramuscularly, intracranially, right? You don't get a response. Unless you bought it from Sigma and it has LPS in it. Right? If you add adjuvant, and there are many different adjuvants you can use, now you get a response. Right? That's non-self. You can do the same with self. If you take myelin basic protein from the brain of a mouse, and you inject it in any one of these ways, you don't get much of a response. If you add adjuvant, you get a response. If you now add something really nasty like pertussis toxin, now you get an autoimmune demyelinating disease that paralyzes the mouse called experimental autoimmune encephalomyelitis. So it doesn't seem to matter whether something is non-self or self. What matters is that you add adjuvant. So I had lots of these kinds of questions, many more than I have time to talk about today as a student, but there were no answers. There wasn't a lot of support for asking these questions, and there was zero support for trying to answer them. So I gave up. I did what we often do as scientists. I stopped asking. And I spent 6 years in Basel at the Basel Institute for Immunology and 4 years in Cambridge as a postdoc, trying to figure out how the immune system discriminates self from non-self. And then I got a job at the NIH. And there was a postdoc in the lab next door named Ephraim Fuchs, who's an oncologist. And he was asking the same questions slightly differently. He was asking, why is it that when we have a tumor that definitely should look foreign because it either has virally encoded proteins or mutated proteins or proteins that were on very early in embryogenesis before there was an immune system and disappeared before the immune system came up. These should look foreign, and yet we don't reject these tumors. Yeah, tumors kill us. And I, by that time, was in old fogey mode. And I patted him on the head and said, there, Ephraim, we all know that tumors are immunosuppressive, having no explanation for why they're immunosuppressive. And unlike me, he wouldn't quit. He kept asking. And we spent the next 2 years arguing, discussing, trying to figure out how the immune system does or does not discriminate self from non-self. And we finally decided it doesn't. 2 years. We came up with the idea that an evolutionarily useful immune system doesn't fight everything in the air you breathe and everything in the food you eat, and it doesn't fight the fetuses you carry or the sperm that gets you those fetuses, all of which are foreign. An evolutionarily useful immune system would be something that fights things that are dangerous. And I'm going to take a slight aside here. I also learned in my career that if something seems evolutionarily stupid, it's probably wrong. Right, so keep that in mind. Those are the kinds of questions you need to ask of your textbooks. We'll come back to a couple. So that took 2 years. So we then had a second problem, which is how does the immune system define dangerous? And that took another year. And it happened to me in the bath, actually. I understand Archimedes. I jumped out of the bath. It's really simple. I don't know why it took so long. Something is dangerous if it does damage. And it's not dangerous if it doesn't. And Ephraim even said, if a virus comes in and makes 2 copies of itself and leaves, it's not dangerous. In fact, you might welcome such viruses because they could bring you genetic information that you wouldn't get otherwise. Okay? So that took another year. And then it took another year to figure out how does the immune system know that something has entered the body and is doing damage. And that happened to me on a sheep field. I was training a sheepdog, and it was summer, and it was hot, and sheep have these woolly coats, so you can't train for very long. And so we had trained for about 10 minutes, and then we were taking a break. And the sheep were at the bottom of this little hill grazing, and there were some trees behind them and a little stream. And my dog and I were just sitting there. She was asleep. And all of a sudden, this sheep came running out of the woods, going, meh, meh. They go meh, by the way, not bah. And the dog woke up and went running into the woods. And I got it. We have cells that are like sheepdogs. They're called dendritic cells. And they live in every tissue we've looked at. And they're asleep, curled up, noses under their tails. Yeah? Until some damage happens. And what was important about that sheep is that it wasn't that the dog had noticed the damage. It was that the sheep had told the dog that there was damage. And so we came up with the idea that a cell that is damaged sends alarm signals. And that it's not until those alarm signals are sent that the immune system is activated. So that explains why the newly lactating breast isn't rejected. It's a perfectly normal tissue. Right? It's not sending alarm signals. It explains for a lot of tumors why tumors are not rejected. We'll come back to that. It explains all kinds of things. In fact, it turns out that if you make that one simple assumption, that an immune response is not initiated unless a cell sends an alarm signal, you can explain almost everything the immune system gets right and almost everything it seems to get wrong, which is pretty good for one assumption. So here's how I think it happens. Some of this is fact, some of this is fantasy. You have cells that are normal. They don't all look like this. That are doing their function, whatever that function is. The lung is breathing, and the kidney is excreting things and making hormones. And in those cells, in those tissues, there are these dendritic cells. And in the skin, for example, each one of these dendritic cells is in contact with between 50 and 500 skin cells. And they're asleep. They're metabolically fairly quiet in G0. Should one of these cells die by normal programmed cell death, it gets scavenged and the innards don't come out. But should it get damaged, say blown apart by a virus, now it will send an alarm signal that will wake up this dendritic cell. Those dendritic cells, when they wake up, this is fact, they can drink their entire volume every hour. They get rid of the water. They keep everything else. And if it's a protein that they've picked up, they break it up into little pieces, and they display it on the surface in these special Schlepper molecules called MHC. Right? They're constantly doing that. Whatever is being made inside is displayed. Actually, it's a window display. It's not 100% display. Just like a store's window display is not 100% of what's inside, but it's a pretty good representation of what's being made inside. Not only do they take these proteins that they've picked up and put them on the surface, they actually migrate to a local lymph gland where they continue to express these molecules. And now T cells, who doesn't know what a T cell is? Oh, good for you. A T cell can come, all right, and it will get what we call signal 1, boringly, by recognizing the peptide, right? And it will get signal 2 from the activated antigen-presenting cell or the dendritic cell. Right? Until that T cell gets signal 2, you don't get an immune response. If it only gets signal 1, it dies. Okay? Most of this is fact. In fact, it's all fact, or at least experimental findings. Okay, so what can you explain by this? So there's a whole list, and I won't have time to do the whole list, but I'll give you at least part of the list, right? You can explain why puberty is not a problem, right? You go through puberty. Hopefully, you don't damage yourself too badly, right? Mothers and fetuses, we'll come back to that. Transplants, you can explain why transplants are rejected and fetuses are not. You can explain why tumors are usually not. You can explain why there are parasites. For example, filaria that cause elephantiasis. You know, they live in the lymphatics in intimate contact with the immune system and the immune cells, and yet you don't usually get an immune response. You can explain why you get graft versus host disease when you do a bone marrow transplant. And you can't explain all of allergy. And this doesn't bother me. And I'll tell you why. Right? The immune system has two questions to deal with when it's faced with something. The first one is, do I respond or not? The danger model is about that. Right? The self-non-self model said you respond when something looks foreign. Janeway's model, which some of you know, says that you respond when something is evolutionarily distant and foreign, like bacteria. The danger model says you respond when something does damage. Right? Once you decide to respond, you have a second question to ask, which is, what kind of response do I make? How do I know to make a killer to catch a virus or IgE to catch a worm? Right? And I think allergy is a problem in that question, that universe. And although I have extended the danger model to cover it, I don't have time to do that today. Invite me back next year. Okay? But there is one part of allergy that I can explain, and that is an old question: why are some molecules allergens and others not? Right? So for example, the house dust mite makes at least 15,000 different proteins. 2 are allergens, DRP1 and DRP2. Right? Cats probably make as many proteins as we do, 50,000 or so, or more, and yet 1 is an allergen. Right? What makes a protein or a molecule an allergen? Right? And the answer to me is dead easy. It's an immunogen because it causes damage, or is packaged with something that causes damage, like bee venom, or it mimics an endogenous or exogenous danger signal. So what are the danger signals? Anything that's inside a cell that is normally not outside a cell can be a danger signal. All it requires is that the dendritic cell have a receptor for it. So some of the ones that are known are DNA, RNA, heat shock proteins, several of the heat shock proteins, ATP. 30% of the innards of a cell is ATP. It's a danger signal. It will activate dendritic cells. So that's one category of danger signals, constitutive molecules inside cells that are not normally out. Right? Another category of danger signal is something that the cell makes when it finds itself infected, like alpha interferon. Infected cells secrete alpha interferon, and that activates the local dendritic cells. Another category of danger signal are molecules like hyaluron. So this is a polymer that lines your blood vessels. And if you get bruised and the hyaluron gets broken down, some hydrophobic parts get expressed, and there's a receptor for that, and that activates dendritic cells. So there's all kinds of them. This is something that's too important to just have as one signal. There's at least 20 now known, and people are still finding more. So back to allergens. What did I say? It either causes damage. So pollen has proteases on the surface that it normally uses to dig down the pistil of a plant to get to the egg at the bottom. Right? That can degrade some of the proteins on your cells. DRP1 is a protease that attacks both lung epithelium and immune B cells. So those are the kinds of allergens that cause damage. Right? You can have an allergen in bee venom, and it's the A2 lipase, I think it's a lipase, that causes the damage, and then you have the other molecules that you can make an immune response to. Right? You have some, I used to think that peanuts were a mimic of an endogenous alarm signal, but I learned in Canada about a year ago that that's not it. So the Canadians have now banned peanut oil from baby cream. It turns out you don't become allergic to peanuts by eating them. You become allergic to peanuts if you're a baby and you have a rashy bottom and your mother or father puts baby cream on it. So you have damage, right? Your skin is damaged. You have activated dendritic cells. And it turns out that peanut oil is a really good oil for creams. So the kids are becoming allergic by having peanut oil baby cream put on them at a time when their skin is damaged. And just as an anecdote, you know, the exception that proves the rule. So there's a couple at the NIH that are immunologists that had a baby that had a lot of rashes, and they knew about this. And so they went to specialty stores and got baby creams using sesame oil instead of peanut. And when their baby was 2, he anaphylaxed in his daycare when they fed him hummus. So he survived, but it just shows you, be very careful of what you put on your babies when they have rashy bottoms. Okay? It's a good idea to feed them whatever you're going to put on them first, right? Make sure they've had some butter before you put butter on their skin when their skin is broken. There's a guy in Nice who has shown that if you feed mice peanuts or ovalbumin, you can no longer make them allergic. Right? So feeding is a good thing. Okay, so I can explain the question of what makes an allergen, but I can't explain why some people respond to it and some people don't. And anybody who wants to ask me about that later can. All right, let's quickly do fetuses, transplants, and tumors. So these are 3 foreign tissues, and you respond to some, and you don't respond to some. Why is that? Why is it that we accept fetuses, and we don't accept transplants, and on the whole, we don't reject tumors? So let's do fetuses first. So what's written in the textbooks? What was written in the textbooks was written first in 1953, I think, by Peter Medawar, who was one of the 2 people who got a Nobel Prize for the self-non-self model. And he understood that pregnancy was a real problem because fetuses are foreign, and yet they're not rejected. And he suggested a couple of things that are in the textbooks that are evolutionarily amazingly dumb. All right? The first one is the fetus immunosuppresses the mother. That's why the mother doesn't reject it. How many of you have carried fetuses? Did you have to live in a bubble? No. Did Medawar notice? Has anybody else noticed? Pregnant individuals are pretty healthy. They're not immunosuppressed. All those cows and pigs and sheep out there, they're not living in bubbles. The idea that you take the individual that's carrying the next generation and turn them into AIDS patients is pretty dumb. And it's not true. Right? There is one condition that has been brought to me several times, and that is that in malaria endemic areas, women undergoing their first pregnancy, if they get malaria, do very badly. They can die, their fetuses die. The fetuses are born with low birth weight if neither one of them dies. And that has been used as evidence that pregnant women are immunosuppressed. It turns out that it's actually evidence that pregnant women can make a great immune response. The problem is these women are 12, 13, 14 when they're having their first pregnancy, and there's a variant of malaria that only grows in pregnant women because it takes itself to the placenta. It has a receptor for a particular chondroitin sulfate that is only on the placenta. And so this woman has never seen this variant before. She's making a primary immune response. And the primary immune response, we know, is slow and weak compared to the secondary memory response. So if they make it through the first pregnancy, they do better on the second and they're fine by the third. And people have gone out into these areas and bled women and men, and they find antibodies in the serum of women who have had babies to this variant, and they don't find it in men. So proof that you can make a primary immune response during pregnancy. Right? Now, there are probably other pathogens are smart, right, that are taking advantage of the pregnant state. And once we figure out how they're doing that, like toxoplasma, then we can understand what the immune system is actually doing. But it's not immunosuppressed. You get flu, you're fine. Right? So the second thing that Medawar suggested was that the placenta, this is also in textbooks, that the placenta is both an immunological and physical barrier between mother and fetus. That's also really dumb, because if your fetus gets infected, it means you can't do anything about it. And it's dead, and so are you. Right? And it's also not true. There's traffic in both directions. Right? We know that there's traffic from fetus to mother, because you can bleed a pregnant woman, you can find the fetal cells using fluorescence sorting, for example. And people do that now, and they isolate them in order to study them for mutants and whatever, to do a prenatal diagnosis. That's traffic from fetus to mother. There's also traffic from mother to fetus. This was first discovered in Leiden at Eurotransplant. Jan van Root was running that, and they have a whole list of people that are waiting for kidneys. And sometimes you have a living donor. You don't have to wait for somebody to die. So you have a living donor. If you have a living donor, they always test the immune response of the recipient against the donor first, because if it's already strong, that kidney's dead in a week, right? No sense doing it. Now, mothers are often donors, and much more often than fathers. And it turns out that about 1/3 of the people waiting for a kidney, when tested for their immune response against their mother, they don't respond against the non-inherited HLA antigens of the mother. These are the transplantation antigens. They respond to their fathers, non-inherited, but not their mother. And Li Zhang and Rick Miller followed that up in Canada with mice, where you can genetically label the father, the mother, and the F1. And they found that about 1/3 of adult mice are microchimeric for the bone marrow-derived cells of their mothers. They're carrying part of their mother's immune system. 1/3 of us sitting in this room are carrying immune cells from our mothers. And if you ever needed a kidney, go ask your mother. Okay? So those have been the explanations for why mothers don't reject their fetuses. Here's the danger model's explanation. As long as that fetus is healthy and developing normally, it's not sending alarm signals. It doesn't matter that it's foreign, like really foreign, right? You would reject a kidney from that baby, But you don't reject the baby because it's not sending alarm signals. Now, should it send alarm signals? Should something happen developmentally? There's a lot of programmed cell death that has to happen in development. If something goes wrong with that, yeah, you might make an immune response, and you might clear it. And that could explain some of the spontaneous immune-mediated rejections that we do see in fetuses. But on the whole, you don't reject your fetus if it's developing normally. Now, I bet that there's at least one person in the audience wondering about Rh disease. Okay, listen guys, ask me these questions. Please, ask me during the talk. If I lose you, you're gone, right? So if you have a confusion or if you know something that doesn't seem to fit, raise your hand. Okay, Rh. Rh positive babies in Rh negative mothers. The Rh negative mother will make an immune response. But it turns out that it's not the pregnancy that causes that. It's birth. Birth is dangerous, right? You have ripping and bleeding, surgeons with knives, right? Alarm signals. And physicians know this because they allow that pregnancy to happen. And then when the woman is nearing birth, or the baby is nearing birth, they give the mother antibodies against the Rh to cover it up. It's not the pregnancy. It's the birth. And then it's the second baby that has a problem. First baby doesn't have a problem. Okay, so the model says that you don't reject fetuses because they don't look dangerous. So why do you reject transplants? You've got a burn patient, you're trying to put a transplant on them, you're trying to help them. Where's the danger? Somebody. It's already happened. Because of the burn. Yeah, absolutely. That's one of them, right? Suppose you take a normal, healthy mouse, and you cut a piece of skin off, and you put another piece of skin on. Where's the danger? Cutting the piece. Surgeons are dangerous, right? They cut, they burn small vessels, they put organs on helicopters and fly them around the country before they get ischemia, they get post-reperfusion ischemia. The organ is damaged, the recipient is damaged. The prediction is that if you could do a transplant without doing damage, it would take. And you can't do it. Even a blood transfusion. You take the blood out, you cool it. That's called a heat shock, and that activates the dendritic cells. You look at dendritic cells cross-eyed, and they get activated. In fact, we grow them from bone marrow, right? 6 days in various hormones. And we discovered that if you harvest them on day 6, and day 6 is a Friday, you have a greater frequency of spontaneously activated dendritic cells than if day 6 is a Monday. Tells you something about the working habits of American graduate students. Okay? The incubator isn't opened and closed as often during the weekend, right? So the temperature doesn't flux, and the pH doesn't flux, and that's enough. That's enough to activate dendritic cells. But if you could do a transplant without danger, it should take, and actually I had a postdoc who did it, and I'm going to very quickly tell you the experiment. So what he did is he took mice that have no immune systems. These are rag knockouts or skid mice. They have to live in a bubble, right? They can't reject anything. And under the kidney capsule, he put a neonatal heart that differed enough that had this mouse had an immune system, it would have rejected that kidney anywhere between 14 and 26 days. But because it doesn't have an immune system, it doesn't reject. He lets the kidney sit for 9 months or 1 month, and then he gives the mouse an immune system by giving it fetal liver cells and allows it to develop an immune system. And he asks, is the heart rejected? Okay. And it turns out that the heart that had been there for 1 month is rejected, and the heart that had been there for 9 months is not. Now, what we know is that, having done it again, by the way, this takes a long time to do these experiments, at 1 month, you still have healing molecules up, like various heat shock proteins, VEGF, and so on, HSP27. Right? That heart has not healed in yet. By 9 months, it's healed in enough that even though it's foreign, the immune system does not reject it. And not only does it not reject it, if you now try to immunize here, right, and then you look later for killer cells against the heart donor, so this is percent kill, and this is a titration of killers. Right? In the mice that were not treated, meaning they never got a heart, right, and you immunize them, you now see good killers. And by the way, if I draw this on a computer, you'll believe me, but if I draw this like this, you won't. In the animals that had had the heart for one, that's true, by the way. I've done that test. Drawn things and asked people, do you really believe the lines look like that? The animals that had had the heart for 1 month also rejected. This is, the dotted line is killing on self. And the animals that had had the heart for 9 months didn't. You don't need to draw this on a computer. It is so obvious. Right? Not only does the heart not get rejected, if you leave it long enough, it tolerizes for itself. Right? The immune system, the T cells are going around. They see the kidney. They see the heart. They get signal 1. They don't get signal 2. They die. Right? Tumors. I have 10 minutes, so I'm going to do tumors in 3. And then you can ask the questions you should have asked earlier. Why don't you reject a tumor? Where are the alarm signals in a tumor? Right? You've got a tumor cell, and it's multiplying or dividing, because in biology that's the same. Right? It's growing. The immune system can't discriminate between rapid cell division in a tumor, rapid cell division in the bone marrow, rapid cell division in the skin, rapid cell division in the gut, which, by the way, replaces itself every 3 days, and the rapid cell division in a tumor. It's a healthy, happy cell that has lost growth control. No alarm signals. Now, eventually, if this thing gets too big and it's poorly vascularized, it'll start to die in the middle by starvation. And starvation death is also programmed cell death. And so still no alarm signals. But eventually one day, maybe, the amount of death in the tumor starts to overload the scavenging capacity. And now, even though the cells are dying by programmed cell death, they don't get scavenged, so they go into secondary necrosis. They fall apart. And they release alarm signals. And now you might get an immune response, because if there is a dendritic cell living in here, it'll get activated, it'll pick up some tumor antigens, it'll go to the local lymph node, it'll express those tumor antigens in MHC, and a T cell going by could see that and get activated. However, what's been happening to the T cells as this tumor has been growing and expressing the tumor antigen? They're getting signal 1 without signal 2 and dying. So you've got 3 left. And what do? Each 3 becomes about 10. And they go out and try and kill the tumor. Each killer cell, it is estimated, can kill between 3 and 30 targets. It takes 3 days, once those cells have exhausted themselves, to go back into the node and get activated again. So you're killing a few tumor cells. What's the tumor doing in the meantime? Growing like crazy. You've lost the race. You had a question? Louder, louder, louder. I'm old.
What is the dendritic cell is doing over there?
Dendritic cells live in every tissue we've looked at.
Yes, but it's not like...
Quiet. It's completely quiet until it gets activated.
No, but it's not like it's being pushed out by the tumor itself, so how can it be inside of the tumor?
So they actually intercalate. So the question is, what is the dendritic cell doing, and why doesn't it get pushed out of the tumor as the tumor is growing? As far as we know, in skin, for example, where the skin is growing constantly, right, the dendritic cells have figured out, I don't know how, to sit there while the cells are going by them. And they're still in contact with them. So they know how to let go, and they know how to hold on. So the dendritic cells in a tumor are the same. And as the tumor is getting bigger, right, more dendritic cells are coming in from the blood, dendritic cell precursors. So as the tissue becomes dendritic cell depleted because it's growing, more dendritic cells come in. And some tumors have more dendritic cells than others. So the ones that don't have many will also be very poor at starting an immune response. So what do you need to do here? What you need to do is you need to initiate an immune response. And you need to keep it going. And here's what happens in almost every paper you read. You've got a tumor that's growing. People do something. They infect it with a virus. They inject it with BCG. They make a vaccine. They do something to prime an immune response. Right? And if they have a good vaccine or a good treatment, the tumor gets smaller. And then it starts to grow again. And so they boost, and the tumor gets smaller. And if they stop, which they usually do, the tumor will grow out and kill the patient. What is wrong with this picture? This is what you see in paper after paper after paper after paper. People prime, they boost, the tumor, and then it comes up. What are they doing wrong? They should have learned what?
Learned by now and not elicited and not abolished the immune response later on. It shouldn't have grown, boosted.
So the tumor will. So if you've gotten rid of the last tumor cell, you're fine, right? But here's what's happening with the immune response. You prime, you get an immune response, it peaks at day 7, it's gone by day 14. You boost, it peaks at day 7, and it's gone by day 14. The cells rest. They're still there, but they're resting killers, right? Because nobody has given them an alarm signal to keep going. The dendritic cells live between 7 and 21 days. You need to reboost it. These people, this is important, so give me 2 minutes.
No worries.
People are in what I call vaccine mode. And what is vaccine mode? You prime and you boost. And it works for a virus or bacterial disease. Because you prime, you expand the number of cells that can see that pathogen. You boost, you expand them again. You can't expand them much more than that because the immune system has homeostatic mechanisms to prevent the entire immune system from going against one pathogen. All right, so you prime and you boost. The cells get activated, and after a while, they go into a resting memory stage. You boost them, they get activated, they go into a resting memory stage. Now, when the pathogen comes back, months later, they do the damage that reactivates the whole system. But the killer killing a tumor cell doesn't do that. That is programmed cell death. They get scavenged. So you prime for a tumor, you activate the killer cells, they're going to kill some tumor cells, the tumor will get smaller. You boost, you activate some killer cells, more of them, the tumor will get smaller. If you got rid of the last tumor cell, you're great, you're home free. If not, if you don't boost again, and again, and again, that tumor is going to beat you. You have to keep boosting. Now, why did I say this is important? It's important because what you think influences what you do. If you remember nothing else from this talk, remember that. 2 things. A, if you have a question, ask it. B, what you think influences what you do. So if you think that once you've activated the immune system against something foreign, it's going to continue to fight that foreign thing until the foreign thing is gone, then priming and boosting is fine, right? But if you think that you need an alarm signal to keep that immune response going, you have to keep boosting. And I have not been able to convince the tumor people to keep boosting, except for one guy in Spain published a paper in the Journal of the National Cancer Institute, and it sank like a stone. But here's what he did. He took people with lymphoma. Wrong marker. And from each person, this is a B-cell tumor, so it has a lot of antibody, and it makes a lot of antibody. They isolate the antibody from each tumor, and they make a vaccine of that. And it works. Okay? And if you prime and you boost, which is what people at Stanford have been doing with this vaccine, you can get people to delay dying from their lymphoma. And what he found was, his name is Ben Dandy. You can look him up, Journal of the National Cancer Institute. What he found is if he kept boosting, he could keep them alive as long as he was boosting. He had one patient in this study that died. And I wrote to them, I said, what happened? He said, we ran out of tumor at 2 years, and he died at 2.5. Now, it's amazing how difficult it is to get people to change their minds. We've been trying to cure cancer for many years. Nixon declared war on cancer in the 70s. I mean, you consider how many dollars and euros and rupees and whatever has been spent on cancer research, and we're not doing much better today than Bill Coley was doing in the 1890s when he was curing 30% of his patients. What you think influences what you do. Thank you for listening.
Thank you, Polly. That was a very amazing talk and a very good way of storytelling and very amusing.
Great, it was amusing.
And I now open the floor for questions. Do we have some questions for Polly?
No?
Yes, one right there. You're good.
Oh, cool. Very cool.
Thank you for the fascinating talk. I have a question. I'm not sure whether it's a good question because I'm
There
not
are
sure
no bad questions.
what's going on. When there's a bone marrow transplant, they register all the donors, and I'm not sure what they're actually testing for, but like they match up donors and recipients. So how does that fit with your theory that transplants get rejected because there's danger signals? Why would you need to like test the donors first? Wouldn't the answer always be the same?
So let's back that question up a little bit. If I pick up a piece of my own skin and put it down, there's danger signals, but it's not rejected, right? If I had an identical twin and took a piece of skin and put it down, it would also not be rejected. So here's why. So my skin has had 71 years to tolerize for itself, right? And my thymus isn't putting out a lot of T cells anymore, a few. Meaning that if a T cell leaves the thymus and it could see skin, and the skin is healthy, eventually, within a few days, it gets tolerized, deleted. So my identical twin skin also works. The reason they match is, remember I said that cells express this window display in MHC of the peptides that are being made inside? So different people have different MHCs. So one of it might look like this. And the structural biologists have discovered that the set of peptides expressed by this cell with that MHC is different from the set of peptides expressed with this MHC, because the groove picks out different peptides. All right? So that's 1,000 antigens, molecules on the surface of a cell that your immune system could see. If you match, that 1,000 is at least something you don't have to worry about. Right? Up to 10% of your T cells could see the peptides in my MHC. So if you can get rid of that 10%, now you're down to a frequency of about 1 in 100,000. Does that make sense? Okay.
Do we have more questions? Could you throw the box right behind you, please?
Thank you for your talk. So,
I have never seen one of those. That is just great.
It's a little bit of
so what
sports.
do you say about the T cell maturation? Is there still like a central and peripheral like negative and positive selection or not? And I mean, I don't remember the name. I think it was AIR. So the enzyme that, or like that,
AIR?
Yeah, AIR. So the cells in the thymus can present like all the different proteins from different cells. So how does that fit into your danger model?
Okay, so this is a real immunologist's question, right? So, so I'm saying that tissues can tolerize for themselves. And people have suggested that that's not necessary, that the T cells grow up in the thymus, and they go through this stage we showed, by the way, that they go through a stage where any molecule they see there, they die. Right? They cannot get activated. So there's this idea that there is this transcription factor, AIRE, A-I-R-E, that helps the cells in the thymus present every antigen that they might be able to, by the way, antigen, I'm sorry, I use that term. Antibody generator, that's where it came from. Any protein the immune system, any molecule the immune system can make a response to, right? So the idea is that any molecule in the periphery would be presented in the thymus, and you delete all the T cells that could see them, right? It turns out not to be true. It turns out that there was a paper in this very obscure journal called the Journal of Experimental Medicine that was completely ignored by the authors of the first paper of AIRE. And here was the experiment. Sorry, it's an immunologist question. You're getting an immunologist's answer, right? BALB/c mice are H2D. So the MHC molecule is D, okay? There are BALB/c mice that don't have a thymus. They're called nude because they also don't have hair. And what these people did is they transplanted into these mice a thymus from B6 mice. Now, BALB/c mice are white. B6 mice are black. And they're H2B. And they differ by lots of things besides skin pigment. Okay. So now all the T cells in this mouse are growing up in a B6 thymus. If AIRE could tolerize them for all B6 molecules, they should accept B6 skin, B6 spleen, B6 thymus, etc. Right? So what's your prediction? What they did is they allowed the T cells to grow up, okay, and then they transplanted B6 skin. Is it rejected or not? It's rejected. They transplanted B6 spleen. It's rejected. They transplanted B6 thymus. Remember, all the T cells had grown up in a B6 thymus. What happens? Is it rejected or not? Who says it's rejected? Who says it's not? Okay, well, you're both right, in a way, because the thymus has two components. It has the epithelium, which was not rejected, and it has the bone marrow component, which was rejected. So what this says is, when you transplant a thymus, remember the bone marrow goes out and dies, right? The thymic epithelium stays. The thymic epithelium can tolerize for thymic epithelium. Punkt. So although a great deal of tolerance does happen in the thymus, because remember, cells in the skin and cells in the kidney and cells in the spleen, they're all doing, the Krebs cycle and making, sodium, potassium, ATPases and all that stuff, right? So many things are the same from cell to cell. But then there are things that are tissue specific. Skin makes keratin, right? For example, right? Brain makes myelin basic protein. Those are not tolerized in the thymus. Now, one molecule that is insulin, and that's been the one that's been used a lot experimentally. And it turns out that developing T cells have insulin receptors. Now, why would a T cell have an insulin receptor? I don't know. But if they have an insulin receptor, you can be pretty well sure that they need to see insulin. Now, insulin levels are things that in the blood go up and down with how much sugar you eat. And wild mice, at least, they don't know where their food's coming from or when. So their insulin levels are probably pretty erratic. And if T cell development needs insulin, you could imagine that a little bit of insulin is being made in the thymus under the control of AIRE so that developing T cells can get the insulin they need. That would be an alternative explanation. Okay? Another question.
Yes. We're running
Throw that thing.
We're running a little low on time, so I think that would be the last question.
Yeah, we've got some student who's next, right? We have to leave room for that.
So,
Yes, but you can always find poly outside on the coffee table or
Go.
something.
okay, how about tumor surgery? Would that elicit an immune response? Have people looked at that? If the
Surgery
immune
of a tumor.
Yes, if the immune system is then activated and maybe kills the remaining cells,
So,
is that known?
if you go into the medical literature, you find anecdotal incidents of when a tumor had had a biopsy taken, that it disappeared.
Yeah, exactly.
Now, that happens less today because biopsies, you know, fine needle biopsy, you stick a needle in, you rummage around and you pull out some tumor, right? But most of the surgeries people do, they try to get all the tumor. I would say don't do that. Take a couple tuned lasers and burn a little corner of that tumor. And then do it again a week later. And then do it again another week later, right? If you take the entire tumor, you will cause danger signals, and the local activated antigen-presenting cells will get activated. But there's no tumor antigen for them to pick up. Right? So don't take the whole thing. Right? I get skin tumors because I'm outside in the sun a lot training dogs, these little basophiliomas. I'm also nickel sensitive. Like 10% of Western women are nickel sensitive. It's because we use 14-karat gold posts when we pierce our ears, and that has nickel in it. And while you're turning the post and causing danger signals, you're driving nickel into your body, and so you're becoming nickel sensitive. Don't do that, use platinum, right? Anyway, I'm nickel sensitive. So when I get one of these things, I get a cheap pair of earrings, I break off the part that goes through my ear, I take the flat piece and I stick it to my nose. And it causes a nice danger, and the tumor goes away. I don't have to go see a doctor. Okay.
Thank you so much, Polly, and everyone else.